Welcome back, ladies and gentlemen, to Biotechnology EOC Review. Let's start with gel electrophoresis. Remember, this is the process used to produce a DNA fingerprint. The way that gel electrophoresis works, we expose DNA to restriction enzymes that cut the DNA at particular segments, and then we insert that DNA into a gel and hook it up to some electricity where the DNA is separated by size and charge. So if I were to insert the DNA here at the top where it said daughter horse, mother horse, and 3 possible fathers, that's where I would insert the DNA, and it runs down the gel. Bigger pieces are near the top because they run slower. Think of trying to travel in a crowd with a large group of people. And smaller pieces run farther down the gel because they're easier to move through that gel. We use gel electrophoresis and DNA fingerprinting for identifying individuals, for either crime scene purposes or, like in the example on here, for paternity. And we also use it for cataloging endangered species. So let's look at this fingerprint over here and figure out who the father of the horse is. When you are looking at a fingerprint that is paternity related, you have to account for the daughter, well in this case the daughter horse's DNA from both the mother and the respective father. It will not match up 100% with either, but if you can match the bands to the mother and the bands that don't match the mother to the father, then you will figure out who the father of that horse is. So this first band right here you see matches that of the mother. It also matches 2 of the fathers. Okay, let's look at the second band the daughter horse has. The horse has this one. You notice it does not match the mothers, but it goes ahead and it matches the father of number 1 and the father number 3. So at this point, we have 3 possible fathers that could be the father of this horse. Okay, this leads us to the next band the daughter horse has. That matches father horse 2 and 3. Now we continue down. The daughter horse gets this band and the one below it from the mother, but could have also gotten the very last one from horse number 3. Now you'll notice that it does match horse number 4, but we can omit that because any of the bands that don't match the mother from the daughter horse, none of them match horse number 4. So we can omit him. So that leaves us with horse number 1, 2, and 3. Now, the important bands to focus on are the ones that do not match the mothers, which would come down to these 2. Now, since horse number 1 only has 1 band that matches the daughters, not the second one, we can eliminate horse number 1. It's the reverse for horse number 2. This bottom one matches the horse's band, but not the top one. But since both don't match, we can eliminate father horse number 2, which leaves us the possible father is going to be horse number 3, because the bands that do not match up with the mothers, those 2 that I've circled, do in fact match up with the daughters. Let's move on to transgenic organisms. A transgenic organism, if you remember, is an organism that has received DNA that belongs to a different species of organism or a different type of organism. The steps in making a transgenic organism, you first have to isolate a target gene. If you look in the picture here, we're trying to insert the fragment for insulin into the bacterial plasmid. So what we have to do is, you'll see right here, this is the target gene. We isolated it. It's no longer a part of the human genome. We then insert it, if you follow the arrow, into this plasmid. It's now called recombinant DNA because we have recombined 2 different DNA from 2 different organisms into 1 genetic ring, the plasmid. We then now take the recombinant DNA and insert it back into the bacterium, where the bacterium will continue to divide so that all of the cells produced through binary fission will contain that recombinant DNA. And when the cell is not dividing, it will undergo protein synthesis, and since it contains a protein that produces that human insulin, that bacteria will then go ahead and produce the product that we want, insulin, which we can then use to treat people with diabetes. Moving on to the Human Genome Project. The Human Genome Project was completed in the early 2000s, and we still use the effects of the Human Genome Project today, particularly in using it to identify individuals that may contain a genetic condition. You can go ahead and get tested and determine if you have a particular mutated gene or genetic condition, and then we can use that information to develop therapy known as gene therapy to help treat those genetic conditions. Stem cells. Stem cells are the ultimate unspecialized cell. They have the potential of differentiating into all of the different specialized cells throughout the body, like brain cells and muscle cells and red blood cells, and they originate in one of two locations. Embryonic stem cells can be found in an embryo, and adult stem cells can be found throughout various organs in an adult. Ethical issues associated with biotechnology. First, we're going to start with stem cell research. Some ethical dilemmas that surround stem cell research have to do with where the source of the cells come from. Is it appropriate to, or ethically okay to destroy future possible life in order to acquire cells to use for the treatment of another individual who is currently alive and potentially save their life? That's an ethical issue. Gene therapy. Gene therapy can be very helpful for an individual that has like cystic fibrosis. However, in the process of using gene therapy, you are inserting DNA into random parts of your genome, which can cause other diseases like leukemia. There's also ethical issues associated with allowing pharmaceutical companies to have patents over certain genes that can prevent the discovery and production of that therapy. And then GMOs. GMOs stand for genetically modified organisms. The ethical issues associated with them: are there health risks to us, the consumer, when purchasing these GMOs? For example, to peanuts. These ethical concerns are very important, and you should be aware of them going into the EOC. If you have any questions, come and see us.